everyone. So so my name is Hayley and I work for MPI and I'm currently working on a couple bee programs, so I'll talk about that today. So primarily the bee pathogen program, but now we're embarking on a new program which I'll talk to you today. And there's some of the people that were involved, and as Kim said, it's been a massive collaboration, this study that we've just undertaken. So I'll talk more about that now.
Always fun, isn't it?
Oh, okay, so before I get into it, I'll just talk about some previous work that was done. So this is kind of the first of its kind. This work was carried out in New Zealand, and it looks very technical, but what this study did was, as Varroa came into the country and as it spread down the country, you'll see the hot spots going down the map, going down to the cooler yellow. So as it took the 6 years to expand down the country, they mapped the viral loads and how the viruses changed as it went down the country. And so our study built upon that, because that's really the only study that's been done to understand pathogen loads in bees in New Zealand. So we thought, how can we build on this study? Yep, and so shortly within starting at MPI, I was shipped off to Canada and the US to look at their bee programs, and they've got quite long-standing bee programs there. There's the National Bee Diagnostic Centre in Canada in a little place called Grand Prairie, and it is a Grand Prairie. And so they've had their bee research lab going for 7 years now, and it's dedicated to just bees. And then there's the USDA. Some of you will probably be familiar with the Bee Informed Partnership, and that's been running for over 25 years. So so we went over and basically absorbed everything that they were doing and then tried to bring it back to New Zealand and emulate what they were doing. And so now I'll talk more about the bee pathogen program. So the bee pathogen program featured 60 apiaries throughout New Zealand. It was a cohort study, so we followed these apiaries, and every 6 months, the likes of Kim would turn up, our bee inspectors would turn up and we would sample them. And so at an apiary, we would sample 8 hives and they would become a composite sample. And you'll see the heat map there on the side. That was from Assure Quality, and that's just hive density. So our study, we sampled 40 apiaries in the North Island and 20 apiaries in the South Island, just to represent these densities. And so what did we collect? So we collected live honeybees, we collected dead or ethanol preserved bees, and we collected a whole lot of data because we interviewed the beekeepers and as well as we inspected the beehives while we were there. And so, yeah, just to recap, I'm not sure, I'm probably in the way there, but it's just a bit of a recap. So we would turn up to an apiary, we would open 8 hives, we would inspect them all, then we would take live bees and dead bees from each hive. They were dead upon collection, ethanol bees and live bees, and they would be a composite sample of 8 hives.
You can move the picture of you anywhere you like.
Oh, okay. So I'll see if hopefully this works. So this is just some sampling in action, and so this is actually us doing it as a demonstration. So this is not an AP2, so we're not as good as Kim at this. So this is just. So a quarter cup of...
Where's your funnel?
yeah.
Where is it?
that's a sauna vinegar top, because we lost our funnel. So we were amateurs trying to do this. So we took the live bees and then we took the ethanol bees. And the reason for taking the live bees is so we would ship them to the lab, and when they get to the lab, they will all be in the same condition, and then we would humanely euthanize them with dry ice and put them in a -80 freezer. And so in theory, the live bees, all of their viruses, bacteria, fungi will still be preserved because the host is still alive and well. The ethanol bees is what we did Varroa mite counts on, so they didn't really need to be in pristine condition getting to the lab. And so, these are our ninjas, our AP2s. We couldn't have done this work without them. So these AP2s, you can see come in there somewhere. So these AP2s actually went out, they had all the relationships with the beekeepers. So we had to do no work on trying to source beekeepers. Our AP2s knew all of these beekeepers already. And Then they went out and did all the work. So they interviewed beekeepers, they took inspections, they took all the sampling for us, and they drove all around New Zealand. I've got some stats to summarise this, but this project, we couldn't have done it without our AP2s. And so when the AP2s were in a hive, so I've just taken a picture, so they would always be looking for problems, but you know, as a reference, they would be looking, so here's just healthy brood as a reference, but they would look for things like deformed wing virus, black shiny bees or chronic bee paralysis virus, American foul brood, everyone's favourite, chalk brood and sac brood, and black queen cell virus, so things that have visible symptoms inside a hive, they were noting any symptoms of that. And so here's just a cool summary of what we've done, so what we've all achieved. So we've got 132,000 bees archived in a freezer at the moment from the project. The AP2s lifted over 2,500 beehive lids throughout the 2.5 years. They shipped 652 bottles and ethanol bee bottles around the country. They did cool things like they spent 1,600 hours looking inside beehives. They drove 51,000 kilometres between apiaries. We hand counted over 16,000 bees in the lab, and we performed over 4,000 diagnostic tests, so either DNA sequencing or through a microscope. And we looked at 15 pathogens and pests. I'll talk about only a few of them today, the results. And you can see down at the bottom, we collected 31,000 data points representing over 100 years of apiary data. So what that means is we've collected enough data as someone who's been beekeeping for 180 years. That's all that means. So we're sitting on this mountain of data that we're currently sifting through. And so this is just a picture from my colleague, Richard, who's not here. He does all of the cool lab stuff. And so the bees come in the boxes, the live bees, and they get chilled down with ice. And then they get put into these tubes. And in this tube down here, there's actually ball bearings. And we put them in this cool machine called a geno grinder, and it turns them into a nice orange juice over there. And I'll move my face, only just there for now. So you can see there's actually, you can do cool things with this geno grinder. So you can put your kids' rattle in there if they're being naughty, and it turns it into a and it turns it into a paste. You can put meat in there and it turns it into flakes. So it's super powerful. So we put these bees in these tubes in with ball bearings, and that just pulverizes them so we can extract all the DNA out of them.
That would be an awesome threat, man. If you don't behave, I'm putting you in that machine.
All right, and so here's some of the pathogens that we tested for. But as I said, we tested for 15 of them. We're still doing testing on them. But we looked at varroa, so we counted the varroa mites. We looked at deformed wing virus, black queen cell virus, chronic bee paralysis virus, cashmere bee virus, Nosema apis, Nosema ceranae, lot maripossum, and Penibacillus larvae. And so when we did all this testing, we would then generate a report for the beekeeper, because it's really important that we have the beekeepers still letting us open their hives and contribute. So they would get a report like this. You see it's sampling round 3, spring 2017. And we'd give them just a bit of a description on, so if they had black queen cell virus, they'd get a plus along this thing. But then we'd just have some notes for their own understanding. So we'd say, yep, you've got black queen cell virus, but it's ubiquitous in New Zealand, it's everywhere. You didn't have chronic bee paralysis virus, it's only found in 4 apiaries in spring in the North and South Island, 2 on each. So it just gave them a bit of context and helping them interpret their results. But then we further gave them a little benchmark of their results. So this is for Nosema ceranae, and you'll see the red dot. You'll see the red dot there, and the red dot is just, it says where that beekeeper is against the cohort. So they can actually benchmark themselves. So that's what the beekeepers got, but then we've got all this data, as we said. And so here's some of the results that I can present today. So this is our varroa mite, and you can see that it's got really nice seasonality, as you'd expect. So in the spring, it's lower. When we're opening beehives in the autumn, it's way up because they've just gone through a big summer with no treatment and they're full of mites. It drops back down, so you get really nice seasonality. You can see in spring we had it around 45%, 50%, and then it would peak up to like 65 to 70% of apiaries in the country that we tested had varroa in autumn. And here's some of the virus data. So the one up the top is black queen cell virus. You can see regardless of season, most apiaries have black queen cell virus. It's almost at 100%. You can see deformed wing virus, quite nice. It kind of mimics varroa. It's got nice seasonality there. You can see cashmere bee virus. In spring, we had a random peak of it where, you know, almost 80% of apiaries had it in spring 2017. And you can see chronic bee paralysis virus. It's quite low, quite rare. You know, only 20% of apiaries 21% of apiaries had it at one point, but then it kind of petered out.
Can I ask you a question?
Yep.
Did you sample hobby beekeeper and commercial beekeeper? Or?
Yep. So yep, we had a collection of beekeepers involved. So yep, commercial and hobbyist beekeepers. And here's some for the nosema and the lotmaria. So the two greens, it's pretty hard to see, but the two greens, darker greens are the nosemas, and the lighter yellowy green is your lotmaria. So they're your microsporidium fungi-like organisms. You can see that the nosemas have the nosema apis and nosema apis and lotmaria have reverse seasonality, which is interesting. So, you know, when we have low nosema apis in autumn, we have high lotmaria or crithidia in autumn. So they're both gut commensals. So at different times, they're operating in the bee's gut. And this is just another thing that we like to produce. So we went around, as I said, and we interviewed all these beekeepers, and these are some of the responses they gave us for floral resources, so pollen or honey. And so the bigger the word, the more common it was. So, you know, most beekeepers are collecting pasture, clover, gorse, everyone's favorite, manuka, kanuka, willow, native bush was some of the most common responses that we got. And as I said, we've got, you know, 180 beekeeping years of experience that we've collected that we're still sitting on. It's a mountain of data, a mountain of research. So we ask things about beekeepers' colony losses. We ask them about their productivity, how much honey, how much propolis, how many queens they made, stuff like that. And we ask them about their management. So, you know, what varroa treatments, when they were treating, how many times they were treating, were they feeding, were they doing brood comb replacement, stuff like that. And because we went and we took GPS coordinates, we have climate data that we can overlay that with. But we also, because we have the GPS coordinates, we can also overlay hive density and how that relates to everything. So, and because we do the lab testing, we've also got all the pathogen loads. So we've got a lot of data to work through and overlay, and what does it mean? And I've put this picture of the Journal of Apicultural Research because in the next month or so, our paper will be published, our very first paper on this. And it's going to be open access, and we will make sure that we send it around to every beekeeping organisation and club in the country. And we've been really we've been really conscientious to make it so it's really digestible and useful for beekeepers. So it's not going to be this really technical study. Our paper that we're about to produce will be something that beekeepers can just read and all comprehend and walk away, because I know a lot of the scientific stuff gets a bit full on sometimes. And so that kind of leads us into our next project, because we're sitting on all this data and we have this responsibility to do something with it all. It's taxpayers funded, and it's got to benefit primary industries, it's going to benefit the beekeeping community. So this new project, we've kind of given everything a rebrand, and we've called it AP Wellbeing. So it's just a more easier package. And so what we're going to do is we're going to develop more tests, because we've still got all the bees in our freezer, if you remember. So we're going to do further testing, and we've got a list there if you're interested in some of the tests that we're going to develop and continue to do. We're also going to create an array of resources and guidelines for beekeepers. And these resources are going to be practical, best practice beekeeping, not scientific papers, as I just mentioned. They're going to be, best practice biosecurity. So we hope to have them out, over the coming years on our webpage. And because we went around, we collected all these bees, we've got this mountain of bees and data that we're sitting on. And as I said, we've become custodians and stewards of all this. So we hope to establish a national collection of this beeson data, and so it's all anonymised, and so other researchers, it might be Plant and Food, it might be Auckland University, it might be Massey University, they can request access to this, so it becomes a future for anybody that wants to undertake research in bees. They can have access to the data. And also what I missed out here was, so in this new project that we're doing, there's a focus on American foulbrood, everybody's favourite, as I like to say. And so we want to do genetic testing on American foulbrood, and we want to do genome sequencing on it, so looking at the genetic diversity of it and what disease types there are, because there's different strains of of American foulbrood. We want to understand what strains of American foulbrood we have in New Zealand, if that has any implications for the management of it. But once we have typing and we know the strain, we can then actually start looking at clusters of disease and how they might be linked to one another. So, you know, beekeeper in Northland might have an outbreak, and then we can show, oh, it's actually the exact same strain as the one in Coromandel. So we can actually start doing tracking or retrospective tracking or real-time tracking of AFB using genetics. So that is something in the next 3 years we hope to do more of. And as I said, resources, I mentioned just earlier, creating an array of resources. So if you just plonk into Google bee biosecurity, the first page you'll get is our landing page, and that's where we've been slowly putting things online. You'll see there's more information on the bee pathogen program, the bee colony loss survey, which is something we also do, the apiculture surveillance program, and now the new api wellbeing project. But there's also bee biosecurity resources there. And one that I'll just highlight, I've brought along today, is BPS ID guides. It's our first output that we've done. So yeah, if there's enough, you're all welcome to it, but you can also download a phone copy if you prefer to have it on your phone. But so there's already a collection of resources there, but this is kind of a cool one to have. So yeah, I can hand those around. But as I said, if you prefer it on your phone, you can download it directly there. And yeah, so if you'd like to get in contact, there's some details there. But yeah, and as I said, I just acknowledge Kim's also working on our new project, going around collecting AFB for us. So we're really grateful for beekeepers like yourselves and AP2s that have done all the hard work. So thank you guys, and thank you for having me. And I've also got...
Thank you very much indeed, Hayley.
Thank you. And I've also
will there ever be a cure for AFB? I mean, are you working towards one? You know, even if it's 5-10 years away,
yeah, I mean that's the $1 million question. I think people are everybody would like to have a cure for AFB, but it's been around. We've had it in the country since as long as we've had bees.
Bee research, that you know of, or?
Well, they used to use antibiotics, so to speak.
Yeah.
They have that mutation of it there. And New Zealand went for the policy of...
No, I know about that, but there's no actual cure coming. No one's actually working on it.
I'm sure like it's stuff like this will probably build understanding, and it might contribute to a cure. But yeah, I'm not aware of any specific research that's being carried out. But I mean, it's been with bees as long as we've had bees. I mean, we've had AFB since when we first brought bees into the country. So it's a entrenched endemic, and it's going to take a long time to fix, unfortunately.
There's a program looking for phages, but I don't know. I haven't heard whether they've had any success. In fact, I heard they hadn't.
Yeah.
That was a year or so ago.
Yeah. So there's lots of like alternative stuff that people are looking at, as you say, like the phages, and yeah. So there's lots of people working on it, and lots of people would like to fix it. But, yeah.
Yeah, Hayley, I've got a question. When there's pathogens in a hive or viruses, we can see bees coming out. Some of them are really obvious, like ring virus, black bees, etc. Is there anything that there's a list of what we can see in the bees that shows us that the bees have got viruses? Because I haven't been able to find this information anywhere.
Yeah, and viruses are hard because that book only features pathogens that have definitive symptoms, but unfortunately for viruses, they look like each other. Really, deformed wing virus and black queen cell virus and possibly chronic beaver paralysis virus are the only 3 viruses that have really distinctive symptoms. Everything else, they just all kind of imitate each other, or you don't see a symptom, but the hive might just dwindle, or other stuff like that.
I mean, I've sort of seen bees come out of the hive and their back legs are actually scratching at their abdomen. So I'm sort of, and that happens quite often, but I've never been able to go, well, that's connected with such and such a virus.
Yeah, and I guess unless you, like I always say to beekeepers, if you see it and you don't know what it is, just report it, because a test will be done. You don't really, with viruses, it's kind of hard. You don't really know until you test it. You look at it, mush all the bees up in that nice juice, and you put them through a fancy machine, and it'll tell you, categorize exactly what it is that's affecting the bees. So yeah, I always just say, if there's anything suspicious, call the hotline, and it's on the little books, and yeah. I've got more information on the project if people are interested in that too, but I didn't print that many, but I can happy to leave these out.
Yep, I've been out front there.
Yeah.
I'd like to say thank you very much indeed
Thank you
for coming along today. A small gesture of thank you.
Thank you.
And good luck with that. Good.